All right, we're recording. So, Kip, David, Hodges, Sipple, Eric, Midkiff, yep, and wearing WVSU stuff. Van Cross, Taylor, gotcha. Michaela, Reed, Redmond, Hemmings, Bowers, Asbury's up there in WVSU gear, Tudor in WVSU gear, Felder, Hudson's here, Burdette, Newton, Massey's here, Richardson, Cedric, Rice, Polly. So we have everybody in person anyway. Got Felder online. Redmond is online. Van Cross is online. Reed is online. Cedric's online. Rice is online. Hodges is online. Excuse me. Burdette is online. And two others, whoever they may be. Let me take a look here. Yeah, oh well. Anyway, let me pull up the PowerPoint. And we'll also start with some announcements. To remind you, unless you get it from me in writing, you will need to be in lab today for the pre-lab lecture. I know I've glanced at my email. Some of you have hours. I plan to go back and check that. So if you haven't received an email from me, you don't need to come to lab today. You still might, because if you've done the video or if you do it like right after class, there's a possibility I'll get it graded in time and you won't have to come. You have a question about that?
Yes.
Okay, good.
Some of them say, like, some of them already have comments or feedback right after I've heard it. But do you still need to look at it?
Yeah.
Okay.
Just to make sure, because if you missed it, like I know there's feedback that should say, hey, you missed this and here's the correct answer or something like that. But yeah, good question. It's very important that you get, that you know exactly what you're getting into tomorrow or next week. Where did it go? Good question. And just so you guys know, I mean, this is, even though we're only doing a pre-lab lecture, this is still, what this is it. You still get graded for it, right? So just by being there, you're getting your 20 points. So for those of you who've done the video and you've got the email from me saying, hey, you don't have to show up for lab this week, you have your 20 points. And for the rest of you who have to show up this week, you will get your 20 points just for being there. So I know it's a little bit of a pain, but in a way, in a sense, it's free points. But it is worth 20 points. It is still a lab. So don't think, oh, it's just a pre-lab, I don't need to be there. You're going to miss out on 20 points if you don't make it. So any questions about that? I think that's all the announcements I have. Does anybody have any questions about any other stuff that's going on? All right, pretty easy. Okay, let's see. If you are, let's just go ahead and start with the first word for attendance. Since it's on here, let's do electron as the first word for attendance. So again, if you're in person, you can send me these words for extra credit if you want, sometime before 9 a.m. If you're watching the live stream, send the word sometime before 9 a.m. And if you're watching the video, unless there's questions attached to the video, then you'll need to send a picture of an electron. Obviously, there's going to be no real pictures of electrons, but if you do a Google image search of electron, something will show up. So anyway, before we jump back into this, let me remind you where we are at. We are doing an overview, right? So we were talking about cellular respiration. Actually, let me back it up a whole lot, really remind you of some stuff. Chapter 5 tells us that cells need ATP to survive, right? That is their form of energy. That's what cells need for energy. Chapter 6 tells us this is where we get it, right? This is how we make that ATP. We make it by breaking down glucose in the presence of oxygen. And in the process, we produce carbon dioxide and water, right? That's what we know about cellular respiration. Then I told you cellular respiration does that in three stages: glycolysis, citric acid cycle, and electron transport. And you're going to need to know those in those order. And you need to know what happens in each one of them, because most of the exam for this portion of it is going to be questions about the three different cycles or stages. So I'll say, in what stage is carbon dioxide produced? In what stage is glucose used? In which stage are high energy electrons stripped away from the molecules? Things like that. And it's going to be a multiple choice exam, and you're going to need to know which stages happens, which stage I'm talking about in the questions. So any questions about that? All right. So then we left off with an overview, right? So I gave you the overview of the three stages. We already talked about glycolysis, which we said you start with glucose, you break that in half, you strip away some electrons, you end up with something called pyruvate. Mitochondria, and we keep breaking down what we started with. And then that comes into the citric acid cycle. We break it down even more. This is where we produce carbon dioxide. Then we get to the third one. And I'll remind you, this is just an overview. So I'm going quick because once I'm done with the overview, then we're going to go back and talk about each step in detail. And that's where we are now, right? We are now finally to the last part. Not of ATP directly, but much more indirectly. Transferring electrons from fuel molecules to a molecule called NAD+. So there's a lot of information in this bullet point. And I would spend time talking about it, but again, this is just an overview. So I'm going to come back to that bullet point later in this presentation, and we can talk about it and break it up, and I can make more sense of it. So for now, I'm just going to leave it at that. One way of looking at it for now is basically this. Right, the whole process of cellular respiration, it's all about making ATP, right? Well, this is the moneymaker. The electron transport chain, the third part, that's where you're making the most ATP. The other two, the glycolysis and the citric acid cycle, they're just there basically to support the electron transport chain. That's the important thing to know. If you keep that in the back of your mind, then that should really help you in the exam. Because when I'm asking about which stage does this and which stage does that, that should really help. So remember, the first two are basically there to support the third one. And then this molecule right here, again, I'm going to talk about this later, NAD+. That's the thing that carries around electrons. And again, this is going to make a lot of thing. It's like, hey, I'm ready to carry an electron or two electrons thing. And then once it gathers the electron, once it gets the electron along with some other stuff, then technically that molecule is called NADH. But again, for now, you don't need to worry about that. I'll talk about it in more detail later. Yeah. So for this slide, what you need to know that we're going to discuss in more detail later is that the first two stages, strip away high energy electrons that are going to be used in the third stage. So any questions about that? All right. So that brings us to the third stage, the moneymaker, so to speak. It is called the electron transport chain, and sometimes it's abbreviated ETC. So again, you need to know that's the third stage, like the final of the three stages. This is where the electrons that were stripped away from the first two processes, they come to the electron transport chain inside the mitochondria. And those are high energy electrons. And those high energy electrons are stripped away of their energy a little bit at a time until finally those electrons are combined with oxygen, and that forms water. And again, I'm going to talk about this in more detail. Test question right there, right? So which of the three stages oxygen used? And the answer is the electron transport chain. And which of the three stages is water produced? And the answer is electron transport chain. Then there's a question that says something like, the electrons come from glucose and ultimately they go where? Forming a molecule of what? So ultimately they go to oxygen, forming a molecule of water. That's another test question. Again, I'm going through it quickly because this is just an overview, and we'll talk about it in more detail soon. Another thing we'll talk about in more detail soon is the fact that the electron transport chain's proteins are embedded in the inner membrane of the mitochondria. Right now, that doesn't mean anything to you, but that will later when we discuss it. But that does bring up another test question. Which of the three stages happens inside the mitochondria? Well, it's going to be citric acid cycle, the one previous to this, and the electron transport chain. Or another way of wording that question would be, which of the three stages happens outside of the mitochondria? In which case that would be glycolysis. Anyway, the electron transport from the NADH, which again, you should just memorize that, is the thing that's just carrying around the electrons. The electron transport chain from NADH to the oxygen releases energy that the cells use to make most of their ATP. In other words, for now, since this is just an overview, what you really need to know is that the first two stages, citric acid cycle, glycolysis and citric acid cycle, they strip away high energy electrons. Those high energy electrons are carried to the electron transport chain, where they go through a bunch of proteins that we're going to talk about later. And every time they do that, every step they make, they're releasing energy because they are high energy. So they kind of lose their energy as they go through this process. And that energy, as we know, energy can't be created or destroyed. So that energy that's lost from the electrons is then used. That's the energy is used to make ATP. And we'll learn how the cells use that energy to make ATP moving forward. So any questions about this slide? Okay, here we go. Now this is still an overview. So the overall equation shows that glucose and oxygen are rearranged to form carbon dioxide and water. I like that. That's the better way of saying it. I usually don't say that. I usually say that, we produce carbon dioxide and water, but I guess it's more accurate to say it's rearranged, right? So back in chapter 2, when we first started talking about chemistry, I made that point, right? Because again, matter is also not created or destroyed. So in this process, we're not creating matter or destroying matter. We are destroying molecules and creating new molecules, but the matter is still the same. We still have the same number of carbons and oxygens and hydrogens on this side as we do that side. But for this, for the exam, that's not important. So again, the main function of cellular respiration is to generate ATP. That will be a question. Someone's going to miss it. I'm going to have a tear in my eye because I keep saying if there's anything that you should remember from chapter 5, it's that cells need ATP to create energy. If there's anything you should remember from chapter 6, which is what we're covering now, it's that cellular respiration is used to create the ATP. Anyway, so yes, that's the whole thing. The whole chapter 6 is about how to create ATP. And again, I've given you this number again. This process can produce about 32 ATP for every molecule of glucose. And I'm not big on numbers, but that is the one you need to know. And you'll notice, like I said, it's about 32. And also you'll notice this picture is not from your textbook because this one says about 38. And I know that can be confusing, but the choices for the multiple choice exam are going to be very big. So it's not like your choices are going to be 32, 36, 38, or 40, right? That wouldn't be fair. It'd be more like, is it 32 or 52 or 22? There'll be big gaps. So try to remember about 32. So any questions about what's on this slide? Okay, it's a good time to remind you, in my opinion, the best way to study this is to look at the big picture and then start adding in details, right? So memorize this equation, not even the numbers, not even the fact that there's, for every one molecule of glucose, it's 6 molecules of oxygen, 6 molecules of carbon dioxide, and 6 molecules of water. You don't even need to know that. But memorize that first, right? Just memorize that respiration uses glucose and oxygen and produces carbon dioxide, water, and ATP. Memorize that. Then start adding in the details, right? And so far, I've given you enough details where you could say a lot more about this. Respiration uses glucose in glycolysis. It produces oxygen in the electron transport chain. It produces carbon dioxide in the citric acid cycle. When it uses the oxygen in the electron transport chain, it produces water in the electron transport chain. Right? All these things I've already taught you, and that we're going to go through in more detail moving forward. Here's a picture of everything we said. Again, just an overview. A lot of test questions here. Again, what's the first? Well, first of all, what are the stages in order, right? Glycolysis, citric acid cycle, electron transport chain. Which of the 3 stages happens outside of the mitochondria? Glycolysis. Which of the 3 cycles happens inside of the mitochondria? Citric acid cycle and electron transport chain. Which of these 2 cycles strips away high energy electrons? Right? Glycolysis and citric acid cycle. Which of the 3 stages uses those high energy electrons that have been stripped away? electron transport chain. Which of the 3 cycles produces ATP? All 3. Which of the 3 cycle stages produces the most ATP? Electron transport chain. Right, a lot of test questions in that one picture. Just an overview. And I think your book's on the same page as I am, where I keep saying, right, memorize the big picture, then start adding the details. I think so with the big picture of cellular respiration in mind, let's examine the process in more detail. Here we go. The 3 stages of cellular respiration. Let's back it up and dig deep. Here we go. Glycolysis. We're back at the first stage, glycolysis. In this process, the 6 carbon what you need to know for the exam. I mean, it won't hurt to memorize. It won't hurt the glucose is 6 carbon. This night, but I'm not going to ask anything about it. Yes, technically carbon, glucose has 6 carbons. And yes, form 2 molecules of pyruvate. And I might ask you that for extra credit, but here's what you need to know. Let's see. You don't even need to know. Obviously, if you have something that has and you have, obviously you're going to have 2 molecules. Each has 3 carbons in it, right? That makes sense, but I'm not going to add, except maybe for extra credit. So you need it for the exam. Again, during analysis, we take glucose, we break it down into pyruvate. Right, that's the first thing you need to know. And that's the first thing I even told you, I think. Monday when I first introduced you to glycolysis, that's what I said. It's a good time to remind you that if you're reading this, maybe in the textbook, but maybe other places either, sometimes pyruvate is called, I'm just going to abbreviate because it's really hard to write with this dumb thing. Yep, see, like that. That's supposed to be a lowercase a. Pyruvic acid is another word for pyruvate. I always use the word pyruvate, and you'll see why later. It's actually to help you memorize things, make things easier later. But for now, just know, if you read pyruvic acid somewhere and like, well, what is that? He never mentioned that. Well, it's the same thing as pyruvate, and we're calling it pyruvate. So in the process of breaking that glucose in half to form pyruvate, we strip away those high energy electrons. Because again, it's all about those high energy electrons. This whole process, well, it's all about making ATP, right? And most of our ATP is made from high energy electrons. So now let's talk a little bit more about this NAD+. NAD+, and I said it earlier, but now I'm going to take a little more time and make sure you understand what it is. This is the molecule that's going to accept those high energy electrons and carry them from one place to the other. And again, like I said earlier, technically it's called NAD+ before it gets the electrons. And then after it gets the electrons, it's called NADH. But I'm not going to test you on that. I just don't want you to get confused if on the test, one of the test questions is going to say something like, which of the 3 stages uses NAD+ to strip away high energy electrons. It's not that you need to memorize it. It's NAD+ and NADH. It's just that I'm using the proper terminology. Because then another test question is going to say, which of the 3 stages uses the electrons from NADH, to produce ATP. So in that way, you need to know the difference between NAD+ and NADH, because I will be using them properly. But I'm not specifically going to say, there's not going to be a test question that says, what's the difference between NAD+ and NADH? But now you know. So again, imagine they're the electron transport chain over there. They're making the most ATP. I give her some glucose. She's glycolysis. And she just breaks it down. And in the process, she's stripping away high energy electrons. They're way over there. She's way over here. I'm NAD+. So I'm like, oh, thanks for those electrons. And I gathered them. So now I'm NAD+ or NADH. I bring them over here, and I'm like, hey, there's some electrons. I get them to them, and they make a lot of ATP with it. That's all it is, right? She's glycolysis. They're electron transport chain, or one of them is, whatever. And I'm just the person or the thing bringing the electrons from one place to the other. So any questions about that so far? We're here. All right. Your book also points out that in this process of breaking that glucose in half to make pyruvate. We're what you need to know is that all the stages produce some ATP. The first two stages produce a little ATP, and the third stage produces a lot. So the number 4, not important. You just need to know that glycolysis produces. If that weren't complicated enough, again, respiration is broken down into 3 stages. And then one of the 3 stages, the first one itself, is also broken down into 2 phases. The energy investment phase and the energy harvest phase. We're going to talk about it because I want you to understand it. But for the exam, I'm not going to ask you to differentiate between the 2. Except maybe for some extra credit. Probably not. Anyway, any questions so far? We're still going to talk about glycolysis, but do we have any questions about what's on here so far? All right. So again, like I said, glycolysis is the one that happens outside of the mitochondria. So that's a test question. Which of the 3 happens outside of the mitochondria? Glycolysis. And again, glycolysis does produce some ATP. So when you get the test question, which of the 3 stages produces ATP? The correct answer is all 3 stages produce ATP. Again, the input is the 6-carbon glucose, right? 1 molecule of the 6-carbon glucose. The output is 2 3-carbon molecules of pyruvate. But more importantly for the exam, you just need to know that we started with glucose, we ended up with pyruvate. So which of the 3 stages uses glucose directly? And that's glycolysis. And it's kind of in the name, right? Glycolysis, glucose. They both start with GL. Which of the 3 stages produces pyruvate? Glycolysis. Any questions so far? Here we go. Back to this whole energy investment, energy harvesting phase. So again, to kick this thing off, it's not technically activation energy, but when you think about it, the concept of activation energy is that, hey, for us to release some energy in this chemical reaction, we need to put some energy in there. Remember, that's what enzymes are. That's what enzymes are for, because they lower activation energy. Anyway, in a sense, that's almost the same, because yes, we are about to get some energy, right? That's the whole purpose, is get some energy by the means of ATP. But in the process, we also have to put in a little bit of energy. And again, I'm not going to ask you about this, but I am going to explain it because it's in the book. So, we broke down that glucose into pyruvate, right? And then that's what this would be. These 3 carbon molecules are pyruvate. And then we add a phosphate group to it. And again, everything I'm telling you right now is not going to be on the exam. I'm just explaining it. Because if you memorize it at this level, then you have no problem under memorizing what's on the exam. So there we go. There's our energy investment phase. We used 2 ATP. We transferred some phosphate groups to those pyruvates. Now we're ready to go. And this is the part that will be on. I know that looks complicated, but what you need to know for the exam is not complicated. What we're seeing here in this picture is high energy electrons have been stripped away. And again, you don't even need to know that that's in the energy harvest phase. You just need to know in glycolysis, we strip away high energy electrons. And again, in the test, I'm going to use the proper terminology. I'm not going to test you on what NAD plus is and NADH. But when you see that, you should think electrons. When you see NAD plus or NADH, mind should immediately think electrons. Because that's what we're using to carry them from one place to the other. Another thing to point out, throughout this exam, there's going to be 2 other molecules that look like NADH. We'll talk about them later, but they're called FADH2. And then we'll talk about the third one when we get there. That's for photosynthesis. But basically what I'm saying is I'm never going to try to trick you in this exam. So anytime you see anything that looks like this, again, whether it's NADH or FADH2 or the one I'm going to teach you later, if it looks like that, your brain should just think, yep, that's the thing carrying around electrons. That's the thing that's going to strip away the high energy electrons and then carry them somewhere else. Because that's another thing, and you'll see when we get there. Photosynthesis also involves stripping away high energy electrons or harvesting high energy electrons. Anyway, so again, if you're studying this, there's so many details you don't have to worry about. Again, you don't need to worry about the energy investment versus energy harvest phase. You don't need to know the detail of the fact that we're adding phosphate groups to the pyruvate. And then notice here, we're adding yet another phosphate group. None of that matters at a 100 level non-biology major course. Fact here is, the important thing for the exam is we've broken down glucose into pyruvate, and in the process, we're stripping away high energy electrons. And then here you can see there's even more steps, 4 of them. Those numbers aren't just random arrows. There's actually different chemical reactions that happen, each with their own enzyme that we don't talk about. But again, we produce a little bit of ATP in the process. Specifically 4, right? Produce here, 2 there. There we go. We have 4. But again, for the exam, you don't need to know the number. You just need to know that glycolysis produces a little bit of ATP. Any questions about this slide? So a lot of detail on there. And remember, if you're studying that, try to remember what I'm saying now. You don't need to memorize all those details, like which one got the phosphate group first, and then how many times did they get an phosphate group added, and how many ATP did we make? Don't worry about that. We made some ATP. We stripped away high energy electrons. That's what's important here. Oh, yeah, and then your book gets even more specific. And you don't need to know this, but I'll really quickly show you because it's in the book. So remember, we have this 3-carbon. At this point, it has 3 phosphates. And this is just showing you how we make an ATP. Well, we're using those phosphates, right? We have a specific enzyme that takes the phosphate off from those molecules I just showed you and attaches it to an ADP, which gives us ATP. So now that's the end result, right? We made a little bit of ATP, and now we have this 3-carbon molecule that still has one of the phosphate groups on it. And again, for the exam, you don't need to know this. All right, any questions so far? All right, let's say the next word for attendance will be right here. I'm pointing at it. I'm not going to say it. So if you're live streaming or if you're watching the recorded video, open your eyes and look at the word. That's the next word for attendance. So if you're in person, send them before 9 a.m. to get extra credit. If you want to, if you're watching the live stream right now, send the words before 9 a.m. to get credit. And if you're watching the video, unless there's questions, then you're going to send me a picture of acid. I said it. Anyway, let's talk about this. So here we go. We started with glucose. We ended with pyruvate, but we're not done yet. And you can look that up if you want. Like, why is it that the pyruvate, like for independent work, why can't pyruvate go into the citric acid cycle? What is it about it that it can't do it? But anyway, for your purposes, just know that it can't. So it has to be groomed. It has to be converted. It has to be changed. You guys know what a coyote is? And I don't mean the animal. Like if you're talking about illegal immigration in the Southwest, does anybody know what a coyote is? No? Well, a coyote is somebody who helps people across the border, like secretly. So basically what I'm going to teach you about is a cellular coyote, right? There's something that helps the pyruvate sneak across the border and get into the mitochondria. And let's talk about it. Whoops. Yeah. Anyway, so here we are talking about grooming the pyruvate because, again, the pyruvate can't cross the border. It's not allowed. So we have to change it. And in the process of changing it to this other thing, and I'll tell you what it is later, it loses a carbon, it's carbon dioxide. So as far as we're concerned, I should have pointed this out earlier, sorry. As far as we're concerned, we are now talking about stage 2, the citric acid cycle, right? We're done talking about glycolysis. We're now talking about the citric acids. Now it's kind of in between glycolysis and the citric acid cycle, right? Because it says right here, citric acid cycle can't use pyruvate. But as far as we're concerned for the exam, these are the same thing. Right? We're talking for, as far as we're concerned, this is the citric acid cycle. So we're including the things we do prior to the citric acid cycle as part of it. Anyway, so we got this pyruvate. We need to groom it in the process. We are losing a carbon as carbon dioxide. You don't need to know where that carbon, or excuse me, you don't need to know that much detail. What you do need to know for the exam is that the Calvin cycle produces carbon dioxide. carbon dioxide? The answer is citric acid cycle. And I think I said Calvin, which is way wrong, and you'll see why later. But yes, citric acid cycle produces carbon dioxide. This is also another time, a good time to remind you that this is also called, depending on where you read it, and I hate writing on this thing, the Krebs cycle. But I don't call it that. It's always going to be called the citric acid cycle, and the reason being is because it's going to help you hopefully not get these questions wrong, because the citric acid cycle produces carbon dioxide, right? So all these C's hopefully will remind you: citric acid cycle, carbon dioxide. And here's a spoiler alert: when we talk about photosynthesis, there's going to be another cycle, and there's going to be a test question that says which of the three cycle uses carbon dioxide, and that is the answer when we get there. So there's a little trick for you in this exam. Any question that asks about carbon dioxide, whether it's using it or producing it, the answer is going to be some sort of cycle, right? So the citric acid cycle produces carbon dioxide. Produces carbon dioxide. And the Calvin cycle in photosynthesis, as you're going to learn later, uses carbon dioxide. So remember, carbon dioxide cycle, carbon dioxide cycle. Anyway, here's what happens. We take that pyruvate, we knock out the carbon from it, and now technically we're left with something called acetic acid. You don't need to know that process. I'm not going to ask you, like, what happens when you take a carbon off of pyruvate? And then, you don't need to know that take a carbon off of pyruvate, you end up with acetic acid. Or there won't be a question that says, where does the acetic acid come from? You won't get that question. You will get this question, though. Which of the three stages is associated with acetic acid? And the answer is the citric acid cycle. Yeah. And here's another thing. Now I can go back and tell you why I don't call pyruvate pyruvic acid, because remember, you can use either word, right? I don't call pyruvate. Any kind of acid, if it has to do with acid, the answer is citric acid cycle. It's in the name, right? So it's all making sense now, hopefully. This is why I don't call it pyruvic acid. This is why I don't call it Krebs cycle. All this to help you remember, all right, if it has an acid, it must be the citric acid cycle, right? I couldn't do that if I called it pyruvic acid, because then you'd be like, oh, pyruvic acid, yeah, that must be formed in the citric acid cycle. But it's not, right? So pyruvate formed in glycolysis, citric acid cycle, you can associate with acetic acid. Any questions so far? All right. We're not over yet, or we're not done yet. Even that acetic acid is not good enough to cross the borders and get into the mitochondria. We have more stuff to happen. And in the process of making the acetic acid, this is also slightly important, we strip away more high energy electrons. And in a sense, hopefully you don't even need to write that down, depending on what you've been writing. But like I said earlier, and I'm basically just saying it again, the first two stages, for the test question, I'm not going to say, you don't need to know that it's when we go from pyruvate to acetic acid that we're stripping away high energy electrons. You don't need to know that. This is going to get more complicated later anyway. All you need to know is citric acid cycle is one of the two stages that we're stripping away high energy electrons. And again, on the exam, NAD+, NADH, FADH2, all those things. When you see something that looks like that, your brain should automatically think, all right, moving electrons. Whether we're stripping them away or moving them or using them, that's all about moving electrons. So each acetic acid is attached to a molecule called coenzyme A to form something called acetyl-CoA. Now, this is really when we get back to that discussion I was having earlier when I was talking about coyotes, right? So again, if somebody's trying to sneak across the border from Mexico to the United States, they need somebody to help them across very often, and that's a coyote, right? The coyote, that person helps them across. And in a sense, that's what this coenzyme A is. Coenzyme A is like, look, I can get you into the mitochondria. Let me help you out. And technically, once that coenzyme A is attached to the acetic acid, right? So here we have acetic acid, here we have coenzyme A. Technically, when you put those things together, Obviously, it's going to have a new name. It's going to be a new molecule. And technically, that's called acetyl-CoA. You don't need to know that it happens in that to the acidic acid, and that produces acetyl-CoA. What you need to know, if anything, is that you might get a test question that says, which of the 3 stages is associated with coenzyme A? Which of the 3 stages is associated with acetyl-CoA? And the answer is citric acid cycle. And that's it, right? Of course, if you're really studious and you're good at these kind of things, if you could memorize the fact that we started with pyruvate, knocked off the carbon, and then we're left with acetic acid, and then we attach that thing to something called coenzyme A, and that forms acetyl-CoA. If you can remember that much detail, then obviously the test will be easier, right? So for studying, if you're good enough at it, yes, memorize all the details. That'll make things easier. And you know yourself better than I do. So if this is really, really confusing you, then I recommend not diving that deep, right? Just learn what you need to learn. Anyway, yeah, so technically everything we said, all right, this is all in preparation for the actual citric acid cycle. So academically, we've been talking about the citric acid cycle, right? Academically, as far as the test is concerned, all this is going to be falling under the citric acid cycle. But technically, what we're talking about is getting things ready to actually go into the mitochondria and actually go into the first reaction of the citric acid cycle. And yes, technically, even though I'm not going to ask you this, but technically, the citric acid cycle actually starts with acetic acid. And you'll see what I mean when we get. The test question will say, which of the 3 stages is associated with acetic acid? And that acid will be your hint that the answer is citric. That one. Any questions on this side? Again, citric acid cycle is one of the 2 stages that happens inside the mitochondria. Everything we've been talking about is basically this little gap right here, right? So the pyruvate left glycolysis, and we lobbed off the carbon and made acetic acid, and then that thing attached to acetyl-CoA, and then it goes in there, right? So that's what we've been talking about, is that little space right here. And technically, if this was more correct, there'd be a little arrow going up and attaching to this, right? Because like I said, we did strip away some high energy electrons. I should write to the publisher. They messed this picture up. Anyway, yeah, here's another way of looking at everything we just said. We started with the pyruvic acid, right? That was the product of glycolysis, that 3-carbon molecule. We lobbed off a carbon, which produced carbon dioxide. We then were left with acetic acid. We strip away some high energy electrons. That's the important part, right? That's what we're after, because the first 2 stages are all about supporting the third one. We stripped away some high energy electrons. We added coenzyme A. Now we have this thing called acetyl-CoA. This thing is now ready to actually go into the mitochondria and start the citric acid cycle. So again, everything you're seeing here is everything I told you earlier. There's no information, no new information there. There are test questions. Which of the 3 stages produces pyruvate? Glycolysis. Which of the 3 stages strips away high energy electrons? Glycolysis and citric acid cycle. Which of the 3 stages produces carbon dioxide? Citric acid cycle. Which of the 3 stages is associated with acetic acid? Citric acid cycle. Which of the 3 stages is associated with coenzyme A and or acetyl-CoA? That would be citric acid cycle. It's a lot of information on this. Okay, so here we go. That acetic acid, right? So we attach to the acetyl-CoA. Acetyl-CoA gets it into the mitochondria. It's now gone, so now we're dealing with acetic acid again. It then joins the actual citric acid cycle, and you'll see why it's called a cycle here in a second. But for now, let's look at the words, then we'll look at it visually. The acetic acid is then dismantled all the way down to carbon dioxide. So again, which of the 3 stages produces carbon dioxide? Citric acid cycle again. So it does it in two different ways. 2 of those carbon dioxide molecules exit as a waste product. I don't even remember why I put a star next to that. If this projector worked properly, I could remind myself. But yes, again, we're producing carbon dioxide. 2 molecules of it. I'm going to put it next to the number 2, not because it's not true, but because I don't require you to memorize that much detail. Again, in this process of stripping down or breaking down this acidic acid, we're producing a little bit more ATP. So again, the test question is going to say, which of the 3 stages produces ATP? A little bit of ATP. But here's the more important part. Like I keep saying, this is so important for the process, not just the exam. The first 2 stages, it's all about stripping away those high energy electrons. So again, NADH, it's the thing, it's got those electrons, it's carrying it off to the electron transport chain. Then there's also this thing, like I said earlier, I kind of hinted at it, FADH2. You don't need to know the difference between the two. When you see any molecule, that's something that's just moving electrons around, right? Whether it's, if it looks, trick you and give you some other word that looks like that. Nope. If it looks like that, it's one of the 3 molecules I taught you, and it is about moving electrons around. And then your book points out that all carbon atoms that enter the cycle as fuel, in other words, all the carbon that is in, all 6 carbons that are in the glucose, right? They are accounted for as carbon dioxide. And then a 4-carbon acceptor molecule that is recycled. I'll put an X through that, not because it's not true, but because what you need to know from this bullet point is what I've already told you, which is that carbon dioxide is produced in the citric acid cycle. Now here's the question I've never asked, and I might do it for extra credit. I guess technically I could ask, ultimately, where did the carbon and the carbon dioxide come from in this whole thing, in all of respiration? Where did it come from? The answer ultimately would be glucose, right? First it was glucose, then it was pyruvate, then it was acetic acid. Either way, it all started as glucose. So all the carbon dioxide, every time you breathe out, that carbon that's coming out of your mouth came from glucose. As far as we're concerned, there are exceptions. We'll talk about it later. So anyway, are there any questions about what's on this slide? All right, I know this isn't the most exciting stuff. I apologize. Not only is it not very exciting, it is also pretty complicated. There we go. Here's a visualization of everything we're talking about. So here's where it all starts off. Like I said, it all starts with the acetic acid, right? This is what we got from the pyruvate, right? So glycolysis was broken down into pyruvate. Pyruvate was then broken down into acetic acid, which then was attached to some enzymes, which came into the citric acid cycle when the enzymes are gone. So here we go. We have the acetic acid in this acceptor molecule, right? And it's a cycle. So they come together to form a 6-carbon molecule of something called citric acid. There's your next test question. And this, please don't ever miss this one. Actually, it's so easy I might not even ask it. Which of the 3 stages is associated with citric acid? And the answer is the citric acid cycle. So anyway, we got this 4-carbon acceptor molecule. It hooks up with this 2-carbon acetic acid molecule. That makes a 6-carbon citric acid molecule, right? Because 4 plus 2 equals 6. Then that thing's broken down even more. We produce some carbon dioxide. We produce a little bit of ATP. More importantly, we strip away some high energy electrons, right? So this is what's important, right? There's a lot of test questions on here. Yeah, and that's it. So anyway, yeah, then once we lob off those 2 carbon dioxides, right? 6 minus 2 equals 4, and then the cycle continues. This is such a easy version. It's a lot more complicated than this. There's a lot, like all these things have names. That acceptor molecule has a name. You can look it up for independent work if you want. The process of this thing attaching to this thing has a name. It all has names. There's so many different things that are not shown on here that you don't need to know, but you could look them up for independent work if you want. Anyway, are there any questions about this slide? All right. Since this thing says citric, and since that kind of looks like a big orange, the next word for attendance will be orange. So if you're in person or watching the live stream, send the word orange before 9 a.m. If you're watching the video that's been recorded, send a picture of an orange if there haven't been questions attached to the video. And I know at this point, some of you are probably thinking, why does he always repeat that? And I know it's annoying, but I promise you, almost every day I have somebody screw it up. And I have to say, no, you're supposed to send it before 9 a.m. So, oh yeah, another good time to remind you, this is an in-person class. I know I put it in writing the other day, but I make it hard. You have to jump through a lot of hoops when you're watching the video or you're live streaming for that reason. We are not done with stage 2. Let's talk about stage 3, the final stage, electron transport, the meat and potatoes, the moneymaker. And before we talk about it specifically, we're going to back up and look at the whole thing, as your book does, right? The detailed book. We're talking about all of cellular respiration. If we're talking about the electrons, and this is a new way of looking at things, as far as I'm concerned, like a new way of me teaching it. And you do need to know this. So not only do you need to know which stage, right? What stage does this happen? That produced. On top of that, you also need to know basically what happens with the electrons. Where do the electrons come from? Where do they go? So that's what we're about to talk about right now. In this entire process, your book describes it as a fall. The electrons fall in a cascade down an energy staircase. Which is basically a better way of thinking about it is we start with these high energy electrons, and as they go from step to step to step to step, they're putting off, right? They're releasing their energy. And that energy is then going to be put to use. And we'll talk about how later. But you definitely need to know that in this whole process of cellular respiration, we start with high energy electrons, and they just lose their energy as they go. It's all about the electrons. And this is important because when we get to photosynthesis, you're going to learn and need to know the difference between the two. That for photosynthesis, it's the opposite. We start with regular old electrons, and then we excite them. We give them energy. And then we put them to use, right? So it's kind of the opposite. Anyway, how many of you have ever jumped a dead battery on a car or seen somebody do it, right? That's what's going on here, right? One person has, probably more. So, you put that one on the red part, you put that one on the black part or middle part of your car, and nothing happens, right? All right, let's put it this way. What happens if you put one red, like on your car that's working, and then the other red on the dead battery? And that's all you've done, right? You've got the reds attached. Is that going to do it? No, right? Because the electrons can't flow, right? Electrons have to come from somewhere and they have to go somewhere. That's basic electronics. Electrons have to come from somewhere, they have to go somewhere. This is why right now, I can put my hand right here and not get electrocuted because the electrons have nowhere to go. Now, if I were to stick a piece of metal in there, the electrons would have somewhere to go and they would go there and I would get electrocuted. But the same thing here. It's the same thing here. Electrons have to come somewhere, they have to go somewhere. And that will be test questions. Where do the electrons come from? So before I move forward, I want you to know this. Ultimately, those electrons, those high energy electrons, came from glucose. The electrons came from glucose. The high energy electrons came from glucose. Right? Because we broke glucose down into pyruvate. We broke that down into acetic acid. The whole time we're stripping away high energy electrons. And yes, technically we took away high energy electrons from pyruvate and took them away from acetic acid. But ultimately, all those things they began as glucose. So where do the electrons come from? They come from glucose. Where do they go? And I'm going to show you this later. I'll visualize it. But the electrons have to have somewhere to go. They go to the oxygen. So they start at glucose, and then they ultimately make their way to oxygen. And that's the story of the electrons. You need to know that. So again, in addition to knowing, which stage does this happen, which stage does that happen, you need to know where the electrons came from, where they went. So they came from the glucose, and they went to the oxygen. And this is really important. And it's also the opposite of what photosynthesis does. Photosynthesis, the oxygen, well, sort of, you'll see what I mean. In a sense, that's where the electrons start, with the oxygen. They don't have any energy. Then we energize them until finally we incorporate them into a molecule of glucose. So it's literally the opposite. Anyway, are there any questions about this slide? Okay. Again, electron transport chain is one of the two that happen inside the mitochondria. All three stages produce ATP. Electron transport chain produces the most. So a few test questions there before we move on. By the time it hits the oxygen, it's as low energy as an electron can be. That energy wasn't just lost, it was put to use. So again, this is everything I said. The electrons came from glucose. Technically, they were stripped away by this NAD+, and then carried around by the NADH. They were taken to the electron transport chain, where finally they came to oxygen and formed water. So I'm going to put it next to these two middle parts, because for the test question, when I ask about where the electrons came from, where they go, this is how it goes, right? The electrons came from glucose, they ended up with the oxygen, and that forms water. You need to know that. And you should associate electrons, oxygen, and water together, because even in photosynthesis, they're going to be together. Because in photosynthesis, water gives up electrons producing oxygen, right? So they're so related, right? If you can memorize for respiration and photosynthesis, in a sense, it should be pretty easy, because I could just say, oh, it's the opposite. That part's just the opposite. Here's a picture of what we're saying. We've stripped away these electrons from the food, right? Glucose is what we talk mostly about, right? That thing's going to carry them away. It strips them away. Technically, once it has the electrons, it's called NADH. It takes them to the electron transport chain, drops the electrons off, at which point it's free to do its job again. It's called NAD plus again. Those things go from one protein to another, which we're going to talk about these three little proteins later. In the process, they're releasing energy, right? These electrons are releasing energy. That energy is used to produce ATP, a lot of it. And we're going to talk about how that happens. And again, those electrons have to go somewhere. Where do they go? They go to the oxygen, and that forms a molecule of water. And look, if you look closely, you'll notice this has 2 electrons, right? I keep saying electrons, but technically it's 2. But you don't need to know that for the exam. Just know that electrons have been used here. So yes, again here, 2 electrons, and then 1/2 of a molecule of oxygen added with 2 protons, right? 2 H+ ions. You don't even need to know that. But here we go. The electrons came to the oxygen, and that forms water. Of course, if you're a thinker, you obviously know that oxygen, just by accepting electrons, is not going to become water. So yes, technically there are other things happening, right? Because where's the hydrogen come from? That is taken care of. But for the exam, I'm not going to ask that much detail. The electrons come from glucose. They go through the whole process. They end up, oxygen is that final electron acceptor. And once it gets it, that forms a molecule of water. So which of the 3 stages uses oxygen? 3 stages uses high energy electrons that have been stripped away, electron transport chain. Which of the 3 stages produces water? Electron transport chain. Any questions? That's a good visualization. All right. Like I already said, there are proteins embedded. You guys know what to do. And again, your video sets the word. The video and send a screenshot of that. That's it. I'll see you guys. Keep an eye on your email. I'm going to go start grading those that you did overnight.